Assalamu alaikum everyone. My name is Hanin Faisal. I'm a second year medical student, and today I'll be giving a path session and I'll be going over embryology. Which topic exactly? It's the first week of development. And if you have any questions, feel free to unmute yourself or write on the chat box. I'll keep it open. And here's my number. If you have any questions afterwards, feel free to text me. I've heard that embryo was a bit tough for you guys, but inshallah, I tried to make the slides as easy as possible or as concise as possible. So let's get started. So today we're going to discuss those objectives. First thing, we're going to talk about ovulation, and then fertilization, then cleavage and implantation, and lastly, we're going to have some practice questions. So before going into the details of the ovulation, fertilization, and so on, we have to build a basic concept so we can build on it. So this concept should be the anatomy of the female reproductive system. Okay, if we zoom into here, this part is called the vagina. And this part is the ovary. Here is the fallopian tube. And here is the uterus. Those four parts are the main parts because each of those parts have different functions. And those are the main four parts. The ovary is important. You're going to know why later. And the same thing with the fallopian tube and the uterus and the vagina. So what you have to know from this slide is just the basic anatomy, which this is the vagina, here's the uterus, this is the fallopian tube, and here's the ovary. Now we have to answer two main questions before going into what's fertilization, because you all know that fertilization happens when the egg fuses with the sperm, right? However, how did the egg reach here to the fallopian tube, which is the site of fertilization, and how did the sperm from the male reproductive organs reach into the female reproductive organ? So those two questions, we're going to answer them now. Firstly, let's answer, how did the oocyte or the oocyte reach to the ampulla? And this is happening by a process called ovulation. So if we look at this picture, you can see that this is the ovary. Let's orient ourselves. This is the ovary. Here's the fallopian tube. And here's the uterus. Okay, in the ovary, you know that the ovary has many follicles. This follicle is called the mature follicle. This mature follicle, it has an egg inside of it, but we don't call it an egg. You have to use the correct medical term, which is called secondary oocyte. So what's the process of ovulation? Ovulation basically happens when this follicle ruptures, releasing the secondary oocyte. So again, what is it? Is when the follicle ruptures, releasing the secondary oocyte. When it releases the secondary oocyte, it will be captured into this fallopian tube. Now we talked about what's the definition of the ovulation. And again, let's repeat it quickly. It's when this follicle ruptures, releasing the secondary oocyte. Now, when does this ovulation happen? Anyone knows? You can type in the chat or... Okay, at puberty. But I mean exactly, you have the menstrual cycle, right? The menstrual cycle, which is exactly at puberty, but I want a specific answer. Like what day exactly of the menstrual cycle? Exactly, 14th day, which is the mid-cycle. So in the mid-cycle, the whole cycle is 28 days. The mid-cycle, which is 14 day, ovulation is going to happen. And we said what's ovulation again? Is when this follicle ruptures, releasing the secondary oocyte. And why does this happen? Like, why exactly on the 14th day an oocyte gets released? Due to a specific hormone. And what hormone is this? Luteinizing hormone. It's very, very important to know this hormone because he might ask you, what's the hormone that stimulated the ovulation? Okay, so let's go over what I wrote. As we said, that ovulation is when the mature ovarian follicle releases the secondary oocyte from the ovary, and this happens at mid-cycle, which, as you guys said, the 14th day, and it's due to the luteinizing hormone. And this luteinizing hormone is stimulating the release of the secondary oocyte, and it's released from the anterior pituitary gland. In the doctor's slides, he mentioned those two clinical names. I don't think it's really important for you right now. You'll take it more in Repro. However, if you see it in a clinical scenario, to understand it, I put it. Mittelschmerz, which is the pain during ovulation. And if there's failure to ovulate, it's called anovulation. Again, just memorize that anovulation is the failure to ovulate, and Mittelschmerz is the pain that happens during ovulation. Okay. Now we're done with ovulation. Before going into fertilization and so on, let's understand the parts of the fallopian tube. Okay, this part is called the fimbriae. This part is called the infundibulum. Here you have the ampulla, and here's the isthmus. And lastly, you have something called interstitium. This interstitium is the one that's closest to the uterus, because here this is the uterus. And here's the interstitium. Here's the isthmus, ampulla, infundibulum, and fimbriae. If I'm going to memorize specific ones, it will be the fimbriae and the ampulla and the isthmus, because those three structures, they're going to have specific functions. Okay, again, we discussed this, but I'll keep on repeating it. What's ovulation? Is when your follicle ruptures, releasing the secondary oocytes into the fallopian tube. Guys, do you know where the fertilization happens exactly? Like what sites? Exactly. Correct. It's the ampulla. Perfect. It happens in the ampulla. However, this oocyte which got released, how can I move this oocyte from this place to the ampulla? There are specific things that will help move it. The first thing is the fimbriae. This fimbriae will help capture the secondary oocytes and move it. Other than that, inside the fallopian tube itself, there is cilia. And you all know what's the function of cilia. It's beating to move something. So the cilia that's found in the fallopian tube will beat to move the secondary oocyte to the site of fertilization, which, as you all said, ampulla. So the second thing is the cilia. And lastly, the last thing you have to know is peristaltic contractions of this fallopian tube. This fallopian tube is made of muscles, and those muscles will contract, helping to move the oocyte to the ampulla. What's the meaning of peristaltic? It's basically if you have something, I can contract at one side, and then I have to relax at the other side, so you can let it move. So think of it like this. I'm contracting here, so I'm pushing it here, but this should be relaxed, because I have to move the oocyte. So it's going to be contracting, here is relaxation, and then when it reaches, it's going to contract again and relax again. So this is the meaning of peristaltic muscular contractions. So again, who can summarize for me the three things that will help move the oocyte? Okay, exactly like Omar said, cilia, fimbriae, and the peristaltic contraction of the muscles. Okay, so now the oocyte reached the site of fertilization. What's the next step? The sperm to reach. Because we said what's fertilization is when there's fusion of the oocyte and the sperm. They fuse together in the size of the ampulla. Now the oocyte reached to the ampulla, and how did we say that happened? By ovulation. Before going into the details of how the sperm reached and so on, let's look closely at the anatomy of the male reproductive system. So this structure is called the testis, and inside the testis, there are some tubules which are called the seminiferous tubules, and this is the part where the sperms get formed. And then on top of it here, we have the epididymis, where the sperms are stored, and this tube is called the vas deferens. Here we have the seminal vesicle, and this is the prostate. It's very important to know those two things, the seminal vesicle and the prostate, and I'll be explaining why in the next slide. Okay, so now we have the sperm, which is formed where? In the testes. How will this sperm reach to the female reproductive organs? Do you think it will be ejaculated as sperm itself, or will it be combined with other things to help the sperm reach there? Exactly, semen. And what is the components? What makes semen? Two things mainly make semen. the sperm itself, which common sense, because that's the thing that I wanted to reach to the female vagina and then go to the uterus and then from there to the fallopian tube. And another thing which is called the seminal fluid. And this is a very, very important concept you guys have to know. This seminal fluid is made from two things. Firstly, the product of the seminal vesicle, and secondly, the product of the prostate. So I said the two important structures that you have to know are the seminal vesicle and the prostate, because both of them release fluids which make up the seminal fluid that's a component of the semen. Now, seminal vesicle is rich in what? First thing is fructose. Any glucose molecule, fructose, any carbohydrate. What's the use of this carbohydrate? Like, why do I have glucose or fructose? Exactly. Provides energy. Exactly, energy or ATP. And why do you think we need energy for the sperm? Exactly, so the sperm can move. Perfect. Exactly. So very important thing is you have to know what's the components of each product, because they might ask you, for example, fructose. Is it a component of the seminal vesicle fluid or is it a component of the prostate fluid? So you have to know that fructose is a component of seminal vesicle fluid. And as you said, it provides energy, because why do I need energy? I want the sperm cell to swim to inside the vagina and then go to the uterus and so on. And another component of the seminal vesicle is fibrinogen. What's fibrinogen? It's something that's going to coagulate the semen after ejaculation. Okay, let me explain it here. It's going to coagulate the semen. Why am I coagulating? Like, what's the benefit of this coagulation? That I'm coagulating the semen, which has the sperm and the seminal fluid. I'm doing this, one thing, to protect the sperm from the acidic environment of the vagina, because the vagina is acidic. I have to protect the sperm. I don't want all the sperms to die, so then what's the use? Because I have to have a sperm that reaches to my fallopian tube. So that's why this fibrinogen is important. It's going to coagulate it as a protective mechanism from the acidic environment of the vagina. And then the second thing is I'm coagulating it to prevent the backflow, because I want to keep it in the vagina. Because if I just have it like as a sperm without the seminal fluids or so on, it can backflow from the vagina. And then there's no use, because we have to have this coagulation to make sure that it stays in the vagina, and then it can go into the uterus, and then reaches the ampulla, and then fertilizes the egg. So now we're done with the products of the seminal vesicle. There are two main products. One is the fructose, and the second thing is fibrinogen. Now let's talk about the products of the prostate. The first thing is citric acid. It's just for nourishment, nothing else. So you just have to memorize that citric acid is for nourishment. Another thing is fibrinolysin. From its name, what does it do? What do you think? Hope you didn't see that. Okay. Exactly, breaks down the breaks the fibrinogen. Why do you think it should break it? Exactly, for the sperm to move again. Perfect, mashallah. So two things. The first thing is going to be the citric acid for the nourishment, and the second thing is fibrinolysin, which is going to liquefy or break the coagulated semen, as you all said, to allow the sperm to move. And lastly, the most important component as well is the sperm. And we said that the sperm is going to be stored where? In the epididymis. When it's found in the epididymis, it's non-motile. It's still not moving. This is something you have to know as well. And other thing, we said that the vagina is an acidic environment, and the sperm moves slowly in this acidic environment while it moves faster in the alkaline environment. That's why it will move faster here in the uterus, because the uterus is alkaline while the vagina is acidic. So it's going to move slowly in the vagina, or it will move faster in the uterus. Okay. Now let's recap again. We said the sperm is formed where? In the seminiferous tubule. And then where will it go? Where will it be stored? In the epididymis. When it's stored in the epididymis, it's non-motile. And then from the epididymis, this tube, it will pass through this tube, which is called the vas deferens. And then it's going to pass through the ejaculatory duct. This is the seminal vesicle. And then it will go to the urethra, and then penis, and then ejaculated into the female's vagina. Okay, how can you memorize this? Easily. I tried to think of a mnemonic, and this mnemonic is 7 up. So let's take it letter by letter. First thing, S will be the seminiferous tubule. E will be the epididymis. V will be the vas deferens. The other E will be ejaculatory duct. N, skip it. U will be the urethra. And lastly, P, which will be the penis. And then it's going to get ejaculated where? In the vagina. So again, the sperm is going to be deposited in the vagina. And we said there is a protein that's released from the seminal vesicle that will help coagulate the semen. What was the protein called? And it's released from the seminal vesicle. Exactly, fibrinogen, exactly. Another protein that's also released from the seminal vesicle that has a similar function is called the vesiculase. It does the exact same function. It will help coagulate it, and so it can stay in the vagina and prevent the backflow. Okay, is everything clear so far? Do you want me to repeat any concept? Do you get the ovulation part and the sperm transport? Okay, I'll explain the last point again. So as we said, let me repeat it again. Okay, so we said in the seminal vesicle, we have a protein, which is called fibrinogen. What does this fibrinogen do? It's going to coagulate the semen. Why am I coagulating the semen? To help it retain in the vagina. Because if I'm not coagulating the semen, the sperm and the seminal fluid and so on, it's going to backflow out of the vagina. Okay, that's the function of fibrinogen, which is released from the seminal vesicle. Another enzyme, which is also released from the seminal vesicle, which has the same exact role, it's called vesiculase. Is it clear? Okay, perfect. So now we talked about ovulation, and we talked about how the sperm gets transported. So, and why do we need all of this? We built this to go through fertilization. Okay, the vesiculase is released from the seminal vesicle, and a seminal vesicle is an organ or a component of the male reproductive system. Okay, perfect. Okay, so again, like a quick recap, we have ovulation, where the follicle is going to burst, releasing the oocyte, and then this oocyte is going to be captured into the fallopian tube, reaching to the ampulla by a lot of mechanisms that will help it. And then we talked about how the sperm will also reach the ampulla. Now let's go to fertilization, which is when this sperm fuses with the secondary oocyte, forming the zygote. But before that, let's describe the structure of the sperm and the structure of the oocyte. So here, this is going to be the tail. We call this the tail. This is the middle piece. And this is the head. The head has the nucleus, which has a very, very important structure, which is called the acrosome. This is really important because it will release enzymes that are really important. And surrounding all of this, surrounding the nucleus, surrounding the mitochondria and the middle piece, surrounding the tail, something called a plasma membrane. Inside the middle piece, there is mitochondria. Why do you think we need a mitochondria inside the middle piece of a sperm? Exactly, ATP, energy. Perfect. We need it for the energy for the sperm to move. Now we finished the structure of the sperm. Let's go into the structure of the oocyte. Let's go from outside. You're going to have layer of cells, and this layer of cells is called the corona radiata. and then you're going to have a jelly coat, which is called the zona pellucida, and then you're going to reach into the cytoplasm, which has the nucleus. So again, a quick recap. The sperm, you have a tail, a middle piece, a head, and this head has a nucleus and an acrosome. While the oocyte, you're going to have a layer of cells, which is the corona radiata, and then you're going to have the zona pellucida, and then the nucleus and the cytoplasm. Before fertilization happens, the sperm has to undergo a specific process to help it move faster and reach to the site of ampulla. And what's this process called? Capacitation. And you really have to know where does it occur, because it occurs in the isthmus, which is this part. This is the uterus. And we know that the sperm is coming from here, which is the vagina. As it goes into the uterus, in the isthmus, there's something which is going to be called capacitation. What's capacitation? Is the removal of the cholesterol molecules and proteins, glycoproteins, that are going to be found on this head. So it's the removal of the glycoproteins and the cholesterol from the sperm head. And why am I removing those glycoproteins and cholesterol? For the sperm head to easily enter to the secondary oocyte. And another function of the capacitation is hyperactivation of the sperm. Hyperactivation means increasing in the motility of the sperm. And that makes sense, because the whole process of fertilization and so on is when this sperm reaches the secondary oocyte. And how can it reach it? It has to swim to it. So that's why we have to increase its motility. And this happens by the process called capacitation. And another thing to help the sperm to help the sperm enter into the secondary oocyte is the removal of the glycoproteins and the cholesterol, which is found in the sperm head. Now let's talk more about fertilization. Fertilization is when this sperm cell fuses with the secondary oocyte, forming the zygote. And this happens in 3 stages. Why does it happen in 3 stages? Because we talked about the oocyte structure. You have the outermost cells are going to be called the corona radiata. This layer is the zona pellucida. And then here's the cytoplasm. For the sperm to enter, it has to penetrate those things. It has to penetrate firstly the corona radiata, and then penetrate the zona pellucida, and then lastly, it has to penetrate the cytoplasm and enter and fuse with the nucleus that's here. So as we said, what were the phases of fertilization? First thing, I have to penetrate the corona radiata. Secondly, I have to penetrate the zona pellucida. And lastly, which is the final step, which is the fusion of this oocyte and the sperm cell membrane. Let's take it step by step. Again, let's orient ourselves. This is the corona radiata. This is the zona pellucida. This is the nucleus. And here's the cytoplasm. Okay, how will the sperm penetrate this corona radiata? We said there's a really important structure that's found in the head of the sperm that releases enzymes. What was this structure called? Acrosome, exactly. This acrosome will release an enzyme which is called hyaluronidase. And this hyaluronidase helps for the sperm to penetrate this corona radiata. So the first thing you have to know in the penetration of the corona radiata is the release of the hyaluronidase enzyme from the acrosome. Other than that, what else there should be for the sperm to penetrate the corona radiata is motility, which makes sense because the sperm has to move so it can pass through the corona radiata. So first thing is the motility for the sperm to move and pass through the corona radiata. And second thing, which is the most important thing, is the hyaluronidase, which is the enzyme that's released from the acrosome, which will help to penetrate the corona radiata. And then finally, the doctor mentioned this in his slides, but I don't think it's that important. However, we said this happens where in the ampulla of the fallopian tube. And in the fallopian tube, there can be mucus secretion, and this mucus secretion can help this process. However, it's not really significant, it's not really important. Just know the motility and the hyaluronidase enzyme. So now we're done. We penetrated the corona radiata. Now we have to penetrate the second layer, which is the zona pellucida. How does this happen? When the sperm will attach to a specific molecule or a specific receptor in this zona pellucida, which is called ZP3. You don't really have to know this ZP3. You'll take it, inshallah, later in Endo-Repro. Just know that it's going to bind to a specific receptor. When it binds to this specific receptor, it's going to initiate a process called acrosomal reaction. To understand this, let's look at the structure of the sperm again. Here's the acrosome, and the thing surrounding it here, this is the plasma membrane. Is there a space between the acrosome and the plasma membrane, or is it attached together? Exactly, there's a space. It's not attached. Because let's see it again. This is the acrosome. And this is the plasma membrane. Here, there's a space. Okay. Now remember this, because we're going to explain it here. When the sperm binds to the ZP3, which is the receptor, just know it's a receptor on the zona pellucida, it's going to initiate the acrosomal reaction. What's the acrosomal reaction? Is when this acrosome binds to the plasma membrane. So we said this is the plasma membrane, and then here's the acrosome. It's going to bind. So there's no space. It's going to bind. And this binding will help the acrosome release enzymes. What's the main enzyme that will help in the penetration of the zona pellucida? It's called acrosin. So now we know two enzymes that are released from the acrosome. The first enzyme was the hyaluronidase, and the second enzyme was the acrosin. Hyaluronidase was important because it's going to penetrate the corona radiata, and acrosin is important because it's going to penetrate the zona pellucida. Is this step clear? Is this phase clear, the penetration of zona pellucida, or do you want me to repeat anything? Okay, perfect. I'll continue. So now the sperm passed through the corona radiata. It passed through the zona pellucida. The final step, the sperm is going to enter into the cytoplasm of the secondary oocyte. However, do you think all the sperm will enter, or is there specific parts that will enter and specific parts that will stay behind? Let's look at this picture and try thinking. Okay. Okay, only some will enter. What will enter and what will not enter? If you look at this picture, what is this? Okay, the head will enter. True. And another thing also will enter. Okay, no, the tail will also enter. Let me explain why. Both the head and the tail will enter, because if you can see here, this is the tail. It's going to enter, and the head also will enter. But something that will not enter is the plasma membrane, which was surrounding them. This plasma membrane, which was surrounding the tail, which was surrounding the mitochondria, and surrounding the head, will stay behind. You can see it here. It stays behind. And the thing that's going to enter is only the head and the tail. And the sperm cell membrane and the mitochondria will be left behind. And with that, we're done with fertilization. We took so far the ovulation part and how the secondary oocyte moved to the fallopian tube. And we talked about how the sperm moved to the fallopian tube. And we finished how fertilization occurs. Is there anything you want me to repeat? I don't mind repeating anything. Okay, does the mitochondria enter? No, the mitochondria will remain behind. The mitochondria will remain behind, and the sperm cell membrane will remain behind. Just know that the head and the tail will enter the cytoplasm of the oocyte. So just those two structures, the head and the tail. Okay, perfect. Okay, now one sperm entered. How are the other sperms prevented from entering? Anyone knows what the reaction is called? Okay, a protective layer. Do you know what's the reaction called? Exactly, cortical reaction. What's basically this cortical reaction? In the cytoplasm of the egg, you have granules, and those granules are called the cortical granules. Those cortical granules have enzymes. What happens is when the sperm penetrates the zona pellucida and enters into the cytoplasm, it will stimulate the cortical granules to burst, releasing the enzymes. And what will the enzymes do? It will make this wall hard. It will harden the wall. If the wall gets hard, the rest of the sperms won't enter, preventing polyspermy. What's the meaning of polyspermy? Let's break it down. Poly means many, and spermy means sperms. So this reaction would prevent the multiple sperms from entering and fertilizing the oocyte. So again, a recap. What happens is you're going to have those cortical granules, which have enzymes. Those cortical granules are going to burst, releasing the enzymes. And those enzymes will harden this wall. And when it's going to get hardened, the rest of the sperms won't enter. Is everything clear so far? Yep. The zona pellucida is the thing that's going to get hardened. The zona pellucida. Okay, what releases the granules? It's a complicated process. Just know that when the sperms enters, it's going to stimulate or send signals for the cortical granules to rupture, releasing the enzymes. Okay, and from the previous session you took, the secondary oocyte, it's stuck at which no, oops, I said it. Okay, it's stuck at which phase of mitosis? Exactly, it's stuck at metaphase 2 of meiosis. So what happens is when the sperm and the egg, when they fuse, they're going to increase calcium levels. And when there's an increase in calcium level, it will cause this egg to resume the meiosis. So it was stuck in metaphase 2 of meiosis. And when the sperm and the egg fuse, it's going to increase the calcium levels. And when the calcium levels increase, it's going to cause the egg to resume this meiosis. Okay. Now we said what were the parts of the sperm that entered. It was the head and the tail. What happens is the tail is going to get degenerated. And then when it gets degenerated, we're just going to have the head, which has the nucleus. And we call it the pronucleus. So this is the pronucleus that came from the sperm. And this is the pronucleus, which is already the nucleus of the oocyte. What happens is now we have two nucleuses. Those two nucleuses, they're going to come closer to each other and form something which is called ooted. And then finally, when they completely fuse with each other, we're going to form the zygote. So we had them as two different pronucleuses, one which came from the sperm and the other one which came from the oocyte. and then they're going to come closer together, forming something which is called ooted. And then finally, when they completely fuse together, we're going to form the zygote. Okay, why do you think fertilization is important? Like, what's the outcomes of those fertilization process? Okay, the formation of the zygote, other than the formation of the zygote. Okay, the chromosome number. Okay, let's explain the last part again. We said that the sperm tail is going to get degenerated, and then it forms the pronucleus. This is going to be the pronucleus of the sperm. And here's the pronucleus of the oocyte. And then both of them will come together, forming together something called ooted. And then when both of them completely fuse together, it's going to be called a zygote. And the outcomes is basically what you said. First thing is going to be the completion of the second meiotic division, because you said that it's going to be stuck at metaphase 2. Then the restoration of the diploid number, because we know that the egg has 23 chromosomes and the sperm has 23 chromosomes. When both of them fuse together, they're going to form a diploid zygote. The meaning of diploid is 46 chromosomes, and haploid is 23 chromosomes. And since we're taking chromosomes from the egg, which is from the female side, and from the maternal side, and we're taking chromosomes from the paternal side, we're going to have a mix of chromosomes, and this causes the variation. So the outcomes of fertilization, first thing is going to be the completion of the metaphase 2. Second thing is when restoration of the diploid number, and lastly is the variation. Now we're done with ovulation. We're done with how the sperm reached the site of fertilization. We talked about how fertilization occurs in 3 stages, which was the crossing of the outer layer, which is the corona radiata, and then the crossing of the zona pellucida, and then finally joining with the female pronucleus, forming the zygote. Now it's cleavage. From its name, cleavage meaning, and this is basically the week 1 of development. Let's start from day 1. Day 1, we have the zygote, which is basically 1 cell. Day 2, this zygote is going to divide, forming 2 identical cells. If it's 2 identical cells, do you think it divided by mitosis or meiosis? Exactly, mitosis. When we have genetically identical cells, it's called mitosis. So this zygote is going to divide to 2, forming 2 cells by mitosis, and this happens in day 2. And then day 3, you're going to have the 4-cell stage, and then day 4, you're going to have a 16-cell stage. And this all happens by mitosis because you're forming genetically identical cells. This 16-cell stage is called morula. You have to know this name. When it divided into 16 cells, it's called morula. Okay, now look at this green thing surrounding the zygote, surrounding the 2 cells, surrounding the 4 cells, surrounding the 8 cells, and the morula. This is the zona pellucida. At what stage did this zona pellucida disappear? Looking at this image. Okay, some people are saying blastocyst and some people are saying morula. Okay, after the morula. It's the blastocyst, because look at the morula, compare the morula with the blastocyst. You can still see the green layer surrounding it, which is called the zona pellucida. While in blastocyst, there's no green layer surrounding it. So in the blastocyst, it's the part where the zona pellucida disappears. And this happens at the end of 4th and the 5th day. Memorize this because he might ask you at which day the zona pellucida disappears. It's the end of the 4th or 5th day. Now it was 2 cells, it divided into 4, and then 6, 8, 16, which is called the morula. And then after that, the zona pellucida disappeared, and blastocyst formed. What is blastocyst? It's also a cells, however, it's more than 16 now. It kept on dividing, and it formed this cell. You're gonna have here cells surrounding, outer, and cells inside. The cells inside, what do we call them? Inner cell mass. From its name, it's the inner cell mass. And then the cells that are outside, we call them as outer cell mass. And inside here, we have fluid. So what's the difference between a morula and a blastocyst? Is fluid started accumulating here. And this is called the blastocele. Here, this is an inner cell mass, and the ones on the outer is going to be the outer cell mass. So by day 5, the morula is going to divide and form this blastocyst, and blastocyst is made of 2 things, the inner cell mass and the outer cell mass. The inner cell mass is called an embryoblast, and outer cell mass is called trophoblast, and the fluid that's inside is called blastocele. So the fluid is called blastocele, and the cells on the outside are the outer cell mass, which are going to develop into a trophoblast, and the cells that are inside are called the embryoblast. And this embryoblast is the one that's going to develop and become the fetus. So a question might come as, which cell is going to develop and form, which group of cells are going to develop and form a fetus? It's going to be the embryoblast. Trophoblast is important for another thing that I'm going to discuss in the next slide, which is basically implantation. Now we have the trophoblast and the embryoblast. This trophoblast again will divide into 2 layers, synthesiotrophoblast and cytotrophoblast. The synthesiotrophoblast is the one that's going to be implanted in the uterus. So this is the uterus wall. And here, this is the blastocyst, which has an inner cell mass, which is the embryoblast, which we said is going to form the fetus. And outside here, this is going to be the outer cell mass, which is the trophoblast. And this trophoblast again will divide into 2 things, the cytotrophoblast and the synthesiotrophoblast. The synthesiotrophoblast is the one that's going to be implanted in the uterus wall. And this happens around 6th day of fertilization. And by that, we're done with the first week of development. And basically, we're done with the whole lecture. We covered the ovulation. We covered how the sperm is going to be transported. We covered the fertilization process in their 3 phases. And lastly, we covered the cleavage and the implantation. Do you have any questions? Is there anything you want me to repeat? Okay, I recap the outcomes. Okay, let's recap the ovulation first, and then I recap with the outcomes. Okay, so ovulation is basically the process where this mature follicle is going to rupture, releasing the oocyte. This is basically ovulation. You have to know that it's when the mature follicle ruptures, releasing the oocyte, and this oocyte is going to be captured by the fimbriae, by the cilia, by the contractions of the muscles of the fallopian tube to move it to the site of fertilization, which is called the ampulla. And this happens at mid-cycle, which is day 14. And why does it happen? Due to the release of the hormone, which is luteinizing hormone. This is literally everything you have to know about ovulation. Who asked the question? Do you get it, or do you want me to repeat? Okay, perfect. Now let's recap the outcomes. Okay. The outcomes of fertilization. First thing, we said it's when the egg and the sperm fuses. And the egg has 23 chromosomes, or the sperm has 23 chromosomes. When 23 and 23, they join together, it's going to be 46 chromosomes, which is the zygote. This is the first outcome you get from fertilization. The second outcome of fertilization, since we're taking chromosomes from the maternal side and from the paternal side, there's going to be a mix of chromosomes, and this causes the genetic variation. So that's the second outcome. And then lastly, the third outcome, we said the secondary oocyte was stuck in metaphase 2 of meiosis. When fertilization happens, this oocyte which was stuck, it will complete the meiosis, and that's it for the outcomes. Okay, what's the name of the fluid? It's called blastocele. Okay, seminal fluid and its components. Do you have a specific question about the seminal fluid and its components for the person that asked? Okay. I think that's it. I answered all the questions. But before we leave, there's some practice questions to make sure that you got the concepts. Let's start with this question. I'll give you like 30 seconds. Write the answer in the chat. If you can write it privately, so we give the other people a chance. Okay, perfect. Everyone is saying D. Yes, the answer is D. Why? Because we said the blastocyst is formed from an inner cell mass and an outer cell mass. Let's look at it. An inner cell mass and an outer cell mass. This inner cell mass is called the embryoblast, and the outer cell mass is called the trophoblast. Now let's solve the next question. Again, 30 seconds. Okay, mashallah, everyone is getting it correct. It's C, the cortical reaction. And what is the cortical reaction again? Quick recap of the cortical reaction. Cortical reaction happens is when the sperm enters. When it enters, it's going to send signals to the cortical granules, which has enzymes, to burst, releasing the enzymes. And then the enzyme is going to harden the zona pellucida. And then when we're hardening the zona pellucida, it will prevent the other sperms from entering. Okay. What hormone stimulates the ovulation in females? Perfect. It's luteinizing hormone. And a follow-up question, at what day? 14. Perfect, mashallah. Then lastly, the last question, which of the following is a component of a fluid that's secreted from the seminal vesicle? Perfect. It's fructose. Why not A? From where is A released? Prostate, exactly. And from where is fibrinolysin released? Prostate as well, mashallah. Correct. You might think the questions are really easy, but I literally got those questions from previous finals. So there's nothing you should worry about. Okay, wait, let me repeat it. Citric acid is a component of the prostate, or fructose is the one that's going to be released from the seminal vesicle, and fibrinolysin is from the prostate as well. So the answer here is fructose. If they ask you about the prostate, it can either be A or D. Okay, finally, some tips and resources for embryo, because a lot of you said that's a bit difficult. For Dr. Paul, watch his recordings, because usually the slides are empty, they're all pictures. So watch the recordings, they're really important. And usually he says underline 10 times. When he says underline 10 times, it's 100% coming in the exam, not even a chance. It's 100% coming. And I linked for you notes, Marwa's notes. They're amazing, amazing, amazing. I used to study from them for embryo, mashallah. It's amazing. Other than that, there is a booklet D&D released, which is called Embryology Essentials. It's also really nice. And there's a video on a channel which is called Pal Bites. It's also nice. And we're done. We finished the whole lecture. Is there anything you want me to repeat? Okay, wait, can I repeat the first question? Let me go back to the first question. Okay, the first question is basically asking you about the components of the blastocyst, which is here. The blastocyst is made from an outer cell mass, which is called the trophoblast, and an inner cell mass, which is called the embryoblast. So if you look at the options, wait, here, trophoblast. Anything else? Yes, I'll send the PowerPoint presentation on the group, inshallah. Okay, for practice questions, use your exam committee. They collect questions from books. You can use that. And anything Dr. Paul says, underline 10 times, and it's 100% coming. So make sure you know it really, really, really well. And I think we're done. Thank you all for joining, and I wish you the best of luck.